I welcome all the registered students in the course of Fundamental of Computational Biology. In previous lecture, you have been gone through the topic Basics of Transcription and Translation. The topic that we will cover in today's lecture is Modeling of Transcription and Translation. Myself, Dr. Kush Kumar Nayak, Assistant Professor, Amity Institute of Biotechnology, Amity University, Chhattisgarh. The contents that we will cover in today's lecture are introduction, central dogma, we will check genetic code, we will see transcription and translation process in very brief. We will also see the ribosomes, genetic code variations, and then check regulation of gene expression. mRNA processing we will discuss in little bit, and then we will move in next part of this lecture, that is models of transcription and translation, types of model, the challenges associated with this, and will at the last we will see future direction. So, let us start with the introduction. Introduction to genetic information. Genetic information refer to the hereditary material that is passed on from one generation to the another generation in living organism. It encompasses the instruction that determine an organism's physical characteristics, growth, development, and overall functioning. The genetic information contained within an organism is stored in its genetic material, which can be found in the form of DNA or RNA. This information play critical role in various biological processes such as reproduction, evolution, and adaptation. The second thing is DNA, gene, and protein. Deoxyribonucleic acid is a genetic material found in most organisms. It consists of two strands forming a double helix structure and is composed of a smaller unit called nucleotide. Genes are segments of a DNA that contains the instruction for building specific protein or performing certain functions within an organism. Proteins are large, complex molecules composed of amino acid. They play crucial role in cell structure, function, and regulations, while gene provide the code or template for the synthesis of protein through a process called protein synthesis or gene expression. This process involves transcription, whereas the genetic information from DNA is transcribed in a molecule called messenger RNA, and translation, where the mRNA is used as a template to access the amino acid into a specific protein. DNA, genes, and proteins are interrelated and work together to determine an organism's characteristics and behavior. Mutation in DNA can result in changes to the genetic information, potentially leading to alteration in genes and proteins, which can have significant effect on an organism's phenotype or traits. So, before moving to the next, let us understand the process of central dogma. Central dogma is a fundamental concept in a molecular biology that outlines the flow of genetic information within a cell. It describes the process by which genetic information is transferred from DNA to RNA and translated into protein form. The central dogma consists three main steps. The first one is transcription, where DNA sequence of a gene is transcribed into complementary RNA molecule. The DNA double helix unwind, and this RNA molecule is known as messenger RNA because it carries the genetic information of DNA to the ribosomes. The second is RNA processing. Before the mRNA can leave the nucleus and translated into a protein, it undergo various modification. This modification including the addition of a protective cap and poly A tail and a removal of intervening sequences called introns are collectively known as a RNA process. This ensures that the mRNA is stable and can be properly recognized by cellular machinery responsible for translation. The third step is translation. Translation is a process by which the mRNA sequence is decoded into a specific sequence of amino acid, leading to a synthesis of a protein. It occurs in a ribosome which read the mRNA sequence in a group of 3 called codons. Each codon corresponds to a specific amino acid or a single to start or stop protein synthesis. The transfer RNA, a molecule which the help of enzymes carry the corresponding amino acid to the ribosomes, where they are joined together to form a polypeptide chain. This chain then fold and undergo further modification to become a functional protein. The central dogma of molecular biology emphasizes the unidirectional flow of genetic information from DNA to RNA and to protein final. Let us understand the genetic code. The genetic code is a set of rules that governs the translation of a genetic information stored in DNA or RNA into the protein. It determines how nucleotide sequences are translated into amino acid sequences, which are then folded into functional protein. The key feature of genetic code includes: first, triplicate codon. The genetic code is read in a group of 3 nucleotides called codon. Each codon corresponds to a specific amino acid or a signal to start or stop protein synthesis. There are total 64 possible codons, but only 20 amino acids and 3 stop signals, which means that multiple codons can code for the same amino acid. Another factor is universality. The genetic code is nearly universal, meaning that the same codon can code for the same amino acid across different organisms. For example, the codon AUG always code for the amino acid methionine, regardless of the species. This universality allows DNA sequence to be deciphered and interpreted by the different organisms. Third factor is redundancy. The genetic code exhibits redundancy or degeneracy, where multiple codons can code for the same amino acid. This redundancy provides a degree of error tolerance in a genetic code, as mutation or variation in a DNA sequence might not always result in a change in the amino acid sequence of protein. The third factor, that is start and stop codons. The codon AUG serve as the start codon, indicating the beginning of protein synthesis. It codes for methionine in the most of organisms. There are also 3 stop codons, namely UAA, UAG and UGA, which signal the termination of a protein synthesis. Fifth factor, it is non-coding codons. Some codons do not code for amino acid, but have specific functions. For example, the codons UAA, UAG and UGA serve as a stop signals, while the codon AUG serve as the initiation codon. Sixth factor is a Wobble base. The third position of codon is known as the Wobble base. The base pairing rules are more flexible at this position, allowing some variation while still coding for the same amino acid. This flexibility allows for the additional redundancy in the genetic code. Understanding the genetic code is a crucial aspect of molecular biology and genetics, as it allows scientists to interpret and manipulate DNA sequences. So, before moving modeling of transcription and translation, let us understand the basics of transcription and translation. Transcription is a vital process in molecular biology by which genetic information encoded in the DNA molecule is transcribed into RNA. As in previous lecture, you know already studied about transcription and translation. It involves the synthesis of complementary RNA molecule using one strand of a DNA template, namely initiation, second is elongation and third is termination. After termination, there is a one more step, that is known as RNA processing. After RNA processing, the processed RNA molecule, which is known as a mRNA, is then ready to be transported out of the nucleus and into the cytoplasm for translation. The translation process, it is governed by which the genetic information carried out. The mRNA molecule is used to synthesize a protein. It involves decoding of mRNA sequence and assembly of corresponding amino acid into a polypeptide chain. The process of translation can be summarized as follows. It can be summarized as follows. The step 1 is known as initiation, step 2 is known as elongation, step 3 is known as termination, and the fourth step is known as a post translation modification. Translation is a complex and highly regulated process. It requires the coordinated action of multiple components including mRNA molecules, ribosomes, tRNA molecules and various protein factors. The major key role that play in transcription translation, that is ribosomes. The ribosomes play a crucial role in protein synthesis as they act as the primary site for translating the genetic information encoded in mRNA into a specific sequence of amino acid. Ribosomes are complex structure composed of ribosomal RNA and proteins. The primary function include first mRNA binding, second tRNA binding, and third one is peptide bond formation. The large subunit of ribosome hold the tRNA carrying the growing polypeptide chain, while the small subunit position the incoming tRNA with its corresponding amino acid. The fourth step that happens as a translocation, where ribosomes facilitate the movement of translocation of tRNA molecules along the RNA molecules during translation. The fifth step is known as accuracy and proofreading, where ribosomes ensures the accuracy of protein synthesis by proofreading the codon anticodon interactions. The sixth step, it is post translation modification. After translation, ribosomes may precipitate in post translation modification of synthesized polypeptide chain. The seventh step, which is known as a ribosome recycling. Upon completion of protein synthesis, ribosome can be recycled for additional round of translation. The another concept that you must know about is the genetic code variation. Genetic code variation refer to deviation or alteration in the standard genetic code, where the correspondence between codon and the encode amino acid is modified. These variation can occur naturally through a genetic mutations or can be artificially engineered in laboratories. Some common type of genetic code variation includes first codon redefinition, second stop codon variation, third codon usage bias, fourth reassignment of tRNA and ARS pairs. The genetic code variation have been observed in various organisms including bacteria, archaea and certain mitochondria and organelles. This variation can have significant impact on protein synthesis, cellular process and even organismal evolution. The regulation of gene expression refer to the mechanism by which cells control the level and timing of a gene expression, or the process by which information stored in genes is used to create functional protein. This regulation is essential for cells to respond to changes in their environment, maintain homeostasis and carry out specific functions. There are several levels at which gene expression can be regulated. The first, at transcriptional level, regulatory protein called transcription factor bind to specific DNA sequences near the genes and either enhance or suppress their transcription. This can be influenced by a signal from the cell or the environment. Second is post transcriptional regulation involves process that occur after DNA has been transcribed into messenger mRNA. These include alternative splicing, where different exons of the pre-mRNA are included or excluded, resulting in different protein products. Third is additionally mRNA stability and transport can be regulated to control the amount of mRNA available for translation. Fourth, the next level regulation is at the translation level, where the mRNA is translated into protein. This can be modulated by the factor that interact with the mRNA or the ribosomes, as well as by the availability of a different amino acid energy sources within the cells. The fifth, which is finally post translation regulation, which involve modifying the functional protein after translation. This can include processes such as phosphorylation, acetylation, methylation and proteolysis, which alter the activity, stability or localization of the protein. mRNA processing is another concept in which refer the series of post translation modification that occur to primary mRNA transcribed before they are fully functional and ready for translation into the protein. This modification are essential for the proper functioning and regulation of a gene expression. There are three main steps involved in mRNA processing, that is known as a capping, second is splicing, and third one is a polyadenylation, which you have been already gone through in previous lectures. So, the second part of this lecture is about the model of transcription and translation. The modeling of transcription and translation refer to the representation of this biological process through the mathematical or computational models. These models aim to simulate and understand the dynamics, mechanism and regulation of a gene expression at the molecular level. Transcription modeling involves mathematical representation of the steps involved in synthesis of mRNA from DNA. This can include the binding of transcription factor to specific gene promoters, the assembly of transcription pre-initiation complex, the elongation of RNA polymerase along the gene, and the termination of transcription. Models may take into account various factors such as concentration of transcription factor, the binding kinetics, and the influence of regulatory elements. Second, the translation modeling focus on the process of protein synthesis from mRNA. It can include the initiation of translation and assembly of ribosomes on mRNA, codon recognition, elongation of the polypeptide chain, and termination of translation. Model may incorporate factors such as mRNA stability, tRNA availability, ribosomes kinetics, and protein folding. The modeling of transcription and translation had provided important insights into how gene expression is regulated, how the dynamics of these processes affect cellular behavior, and how dysregulation can lead to disease. It has been also observed and used in synthetic biology to design genetic circuits or engineer cells with desired gene expression profiles. However, it is important to note that these models are simplification and approximation of a complex biological processes, and their accuracy depend on the quality of data and the underlying assumptions. Let us understand the various type of model used in transcription and translation. There are several types of mathematical model that have been used to study the transcription and translation. These models vary in complexity and level of details they capture. Here are the few common types of this model are: first, deterministic model. This model use ordinary differential equation to describe the average behavior of system over time. They assume well mixed population and do not consider statistics or randomness. Deterministic models are often used to study the dynamic of transcription and translation in a population of a cell. Second, stochastic model. Stochastic model take into account the inherent randomness and fluctuation in gene expression. This model use stochastic differential equation or stochastic simulation algorithm to capture the probabilistic nature of transcription and translation. They are useful for studying noise, variability, and heterogeneity in gene expression. The third category is known as rule based model, which often implemented using agent based model describe individual molecular entities and their interaction. These models simulate the behavior of each molecule and their role or reactions. Rule based model allow for detailed spatiotemporal modeling of gene expression and can capture molecular crowding and other spatial effect. The fourth category is known as the Boolean models, which use binary variables to represent the on off state of gene or regulatory elements. These models are based on the logic rules and describe gene regulatory network. Boolean models have a simplified representation of a gene expression, but can provide insights into the dynamics of regulatory interaction and identify stable gene expression states. The fifth model is known as thermodynamic model, use principle of statistical and mechanics and thermodynamics to describe the binding interaction between DNA, RNA, protein and other molecules. This model incorporate thermodynamic parameters such as binding affinities and free energy to predict the binding and dissociation rate of transcription factor and other regulatory molecules. Let us try to understand the two main methods that being used in the modeling of transcription and translation. The first model is ordinary differential equation, which is known as a ODE model. This model are mathematically model used to describe the dynamic of a system over time. In the context of a gene expression, ODE model are commonly used to represent the dynamics of transcription and translation process. The ODE model are based on differential equation which express how the rate of change of variables depend on the value of those variables themselves. In the case of gene expression, the variables typically represent concentrations or abundance of a different molecular species such as mRNA, protein and transcription factors. The general form of system of ODE representing gene expression can be written as dx by dt equal to function of x and t, where dx by dt represent the rate of change of the variable x with respect to time and can be function of x and t. The f, x and t represent mathematical function that describe the interaction and kinetics of the system. To construct a ODE model, researchers must determine the functional form of f, x and t based on experimental data, knowledge of the biological system or assumption about the underlying mechanism. This function usually incorporate terms that represent product, degradation, binding, unbinding and other relevant processes. The ODE model offers several advantages for describing gene expression dynamic. They can provide insight into the temporal evolution of variables, capture the effect of parameter changes or perturbation and identify steady state or stable solution. There are numerous examples of ODE models applied to the study of transcription and translation process. Here are the few prominent models who work under this ODE category are: first, the ODE capture the dynamics of a protein synthesis, degradation and activation through phosphorylation and dephosphorylation reactions. The model help explain the periodicity of cell cycle events. Let us represent the protein concentration as a P function of time t, and ODE equation for the Koshland models can be written as given in a slide. Another method is known as a Rosenfeld model. This model focuses on a gene transcription and regulation. It incorporates the binding and unbinding of transcription factor to gene promoters as well as the synthesis and degradation of mRNA. The ODE mimic the competition between transcription factor, allowing prediction of gene expression level and understanding the regulatory network dynamics. Let us consider a single gene and represent its mRNA concentration as m function of time. Then ODE equation of Rosenfeld model can be written as given in the slide. The third model, that is known as a Elvis model. This seminal model investigates the dynamic of gene expression noise employing a stochastic ODE hybrid approach. The ODE describes the average behavior of mRNA and protein abundance over time, while stochasticity is incorporated to model the inherent randomness in gene expression. This model helps uncover the mechanism underlying gene expression variability. Let us consider one gene mRNA concentration as m function of time and protein concentration as a p function of time. The ODE equation for Elvis model can be written as given in the slide. The fourth model, that is known as a Osbudek model. This model explores gene expression regulation by transcription factor, specifically the temporal dynamics of gene activation. The ODE capture the protein-protein interaction and transcription factor binding kinetics to their target genes. This model provides insight into the temporal ordering and timing of a gene expression event. Let us consider one gene's mRNA concentration as MT and the concentration of a activated protein as a AT. The ODE equation for the Osbudek model can be written as provided in the slide. The fifth model, that is known as the Gibson model. This model investigates translation regulation by small non-coding RNA such as microRNA. The ODE describes the interaction between mRNA, microRNA and protein. The model demonstrates how microRNA can modulate protein expression level by binding to target mRNA and inhibiting translation. Let us consider one mRNA concentration as MT and concentration of a microRNA as a RT and the concentration of protein as a PT. Then the OD equation for this model can be written as given in the slide. The another model that is used for the study of transcription translation dynamic is stochastic model. The stochastic model for transcription translation are used to capture the inherent randomness and fluctuation in gene expression processes. Unlike deterministic models which assume well mixed population and deterministic rate of reaction, stochastic model explicitly consider the probabilistic nature of molecular interaction and account for the effect of noise on gene expression. These models are particularly valuable when studying systems with low copy number of molecules where stochastic effects play a significant role. There are different type of stochastic model used in the study of transcription and translation, including the first, stochastic differential equation. The stochastic differential equation are a common modeling approach for capturing the fluctuation in gene expression. These equations describe the time evolution of variable considering both deterministic rate of change and stochastic noise. The SD are often used in model gene regulatory network and capture the effect of molecular noise on gene expression system. Second is chemical master equation. The chemical master equation is a fundamental stochastic framework for modeling chemical reactions in biochemical systems. It provides a probabilistic description of the time evolution of probability of a different molecular species, taking into account the discrete nature of molecular counts and stochasticity of reaction events. The CME is commonly used for modeling gene expression and can be numerically solved or approximated using techniques such as the stochastic simulation algorithm or Gillespie algorithm. The third model is stochastic simulation algorithm. This stochastic simulation algorithm such as Gillespie algorithm are widely used for simulating gene expression dynamics. This algorithm explicitly simulate individual molecular event such as transcription and translation based on their respective reaction rate. By randomly selecting reaction event and updating molecular count, stochastic simulation algorithm generate time trajectories for molecular population, providing statistical information about the variability and distribution of gene expression. So, now just move towards the application and example of stochastic modeling, that is gene expression noise. Another area where it is used is bursting in gene expression. The third point where it is used is stochastic transcription. The fourth point is stochastic translation and ribosomes dynamics. The fifth, where protein folding and aggregation with stochasticity can be studied. So, let us discuss about certain challenges and limitations that are associated with the particular topic. That are the various challenges and limitations associated with these topics are complexity of a nucleotide sequence, second, gene expression regulations, third, alternative splicing, fourth, post transcriptional modifications, fifth, gene regulation by non coding RNA, sixth, ribosomes dynamic and protein folding, seventh, accounting for cellular context, eighth, data availability and integration, ninth, model validation and prediction, and 10th, evolutionary consideration. These are the certain points where challenges and limitations can be taken in count. The future direction are as follows: first, integration of multi-omics data, second, advances in single cell analysis, third, incorporating structural information, fourth, enhanced modeling of non-coding RNA, fifth, deep learning and machine learning approaches, Sixth, integration with gene regulatory networks. Seventh, modeling in the context of disease and development. Eighth, advancement in synthetic biology. Ninth, model based drug discovery. 10th, emphasizing reproducibility and open science. These are the certain list of references which can be further used for the more detail.